Back to chapter 23, we're going to start looking at G-protein coupled receptors. Again, a few videos in this series, so let's get going. The G-protein coupled receptors, or the GPCRs as I'm going to refer to them and as they are usually referred to, they're named like that because when a ligand binds, it causes a change in the receptor's conformation that activates a particular G-protein. So a G-protein is a guanine nucleotide binding protein. So that is GTP or GDP as far as we're concerned. It's obviously GMP, but we're not worried about that. GTP or GDP are the guanine nucleotides bound by G proteins. Once a G protein, an activated G protein is produced, it then is able to go and bind a target protein, which in some way alters its activity and elicits a signal transduction cascade or turns off a signal transduction cascade. So we'll come back to that in a little bit. Now, whatever G-protein linked receptors, whatever GPCRs you look at, they all have similar structures, but they can have very, very different amino acid sequences. And this speaks back to the idea that amino acids are chemically and structurally conserved in some cases, and so you can substitute different amino acids out while still preserving an overall structure. So the receptor has 7 transmembrane or alpha helices. So here they are, 1, 2, 3, 4, 5, 6, 7 helices. So it's a 7-pass transmembrane protein. So you can think about how this would have been inserted into the ER lumen membrane, to the ER membrane, to have been delivered to the cell surface. So you can start to put these big pictures together, which you couldn't do with general biology, I feel. So you can think about the translation initiation of this in the cytoplasm, the targeting to the ER membrane, and then the insertion into the ER membrane, and then transit through the endomembrane system for this protein to be deposited in the cell membrane. So you should be able to get a bigger picture of what's going on in cells now. Okay, so what we're going on here. You've got these 7 transmembrane helices, and then you've got these exterior loops, or these cytoplasmic loops, linking the alpha helices together. And there's a region down here in loops C3 and C4 towards the carboxy terminus, which are involved in actual interaction with the G protein. And then the exoplasmic face here, the exterior face here, that right around, where is it? Right in here is where you're going to see the ligand binding on the extracellular face. So we've got G protein activation down here on this side and ligand binding up here on the exoplasmic face. And so the structure of this ligand binding site is going to vary receptor to receptor, because obviously they're going to have different ligands. The receptors are specific for their ligands. So you can regulate these receptors in a variety of ways. A common one is via phosphorylation of amino acids in these cytosolic domains. That's carried out by G-protein linked receptor kinases, and they can act on activated receptors. For example, protein kinase A is activated by G-protein mediated signaling, but then pKa can go back and phosphorylate amino acids on the receptor and therefore inhibit and turn it off, and so that gives you an idea of a feedback loop. You start a signal transduction cascade, you elicit a response, but then the actual part of the response is to turn off the signaling pathway, and you only keep it turned on if the ligand is present. So that's a nice example of negative feedback in signaling pathways. So let's take a look at the G protein and the interaction with the guanine nucleotides and with the receptors. So we're talking about heterotrimeric G proteins. So that is obviously 3 different subunits in the G protein. So here is the trimeric or heterotrimeric G protein. And we're looking at a GS protein here. And there are 2 types: GS, that's a G protein which is stimulatory, and GI, which is a G protein which is inhibitory. So we're dealing with the stimulatory form of the G proteins. 3 subunits: G alpha, which is the largest subunit, and then a beta subunit here, which is linked to the membrane, and then a gamma subunit. So the beta gamma are the 2 small ones over here. Beta gamma stuck together and gamma attached to the membrane there. Okay, so we've got three parts here, alpha, beta, and gamma. So when it binds, so what we're going to have happening here is that in just a resting state with no ligand binding to the receptor, we've got the G protein anchored to the membrane. We've got some kind of effector molecule here, so that's going to elicit the kind of the transduction part of the signaling pathway. Here's a receptor that's inactivated. Here comes along a hormone. A hormone binds to the receptor. That causes a conformational change in the receptor, and that will allow the alpha subunit, this little structural piece of the alpha subunit of the heterotrimeric G protein, to interact with the receptor. So that's the conformational change in the receptor which activates the receptor, and that allows the G protein to stably dock. Now, you got to think about a membrane, a fluid, semi-fluid membrane. These things are always bumping into one another. But if this is the G protein, it bumps into the receptor when the receptor's not activated, it's just going to bump and then dissociate. So they're just bumping into each other. Now, if you change the structure of this by being activated, now when this G protein comes close and touches it, now we get a tighter interaction. And so when we get that tighter interaction between the G protein and the receptor, is what happens is the GDP is released from G alpha, and we have GTP binding to G alpha. And that GTP binding causes the release or the separation of G alpha from G beta gamma. And then G alpha in the GTP bound form can bind this effector and turn that effector on. And then after a short amount of time, the GTP gets hydrolyzed back to GDP, and we go back to the resting state, and the effector is switched on. So the effector here can only be switched on when G alpha is dissociated from beta gamma, and when it's only in that state when GTP is bound. So that's kind of the whole cycle. So we've got this active state with GTP bound and an inactive state with GDP bound. And all we're doing is switching between the two. And that's going to depend on whether the receptor has ligand bound or not. So now, the G protein activity in terms of GTP hydrolysis isn't fantastic always. And so you can regulate the GTPase activity, that just means the hydrolysis of GTP to GDP, by a regulatory protein called a GAP. And a GAP is a GTPase activating or a GTPase accelerating protein. Now, this piece of art is accelerating. I learned a GAP was a GTPase activating protein. And so is what the GAP does is they stimulate the inherent activity of the GTPase so that the GTP is converted to GDP, and we get switching to the off state in terms of the G protein. So this would be G alpha here. So you can regulate GAP abundance and GAP localization to control this switching off. So if we wanted to keep the G alpha active longer and stimulate the pathway, we could inactivate the GAP. Now, we can also regulate the kind of the other side of the process, if you like. We can have an enzyme, a GEF, a guanine nucleotide exchange factor, which helps the G alpha subunit release the GDP and release it and replace it with GTP. So that's called a GEF. So if we want to speed up the switching on of the pathway, then we could stimulate an increase in abundance of the GEF. So on this side here, we're stimulating the turning off, and on this side here, we're stimulating the turning on. So what do these G proteins do? So the most important G protein function is to stimulate the release or formation of second messengers. And so we'll look at cyclic AMP, but we could be talking about calcium, IP3, and all kinds of things. So, for example, the alpha subunit of activated G proteins interact with proteins called adenylyl cyclases or an enzyme called phospholipase C. So you've probably heard about adenylyl cyclase. That's involved in the production of cyclic adenosine monophosphate from ATP. Phospholipase C is involved in cleaving a lipid, and the cleavage products are then involved in the signal transduction pathway. So in addition to the alpha subunits activating signal transduction, beta gamma can also be involved in some systems. So this is the big problem with cell signaling, is that there are lots of variations around general themes. So I'm trying to focus on general themes, not get into the weeds too much with a million examples, which you'll only forget. We'll look at a couple of examples for something concrete. So for example, Gprotein receptor kinases can be activated by beta gamma, so that's one way in which you can use beta gamma, or the acetylcholine, the muscarinic acetylcholine receptor can also stimulate this G protein here, and when it stimulates this G protein here and alpha binds GTP, beta gamma can be released, and beta gamma can act on this ion channel to stimulate the movement of ion channels down a gradient. So that's an example of a Gprotein coupled receptor binding to its ligand alpha, switching out GDP for GTP, and beta gamma going and doing something. And usually we focus on alpha doing something, but this is one example when beta gamma is doing something. So when acetylcholine is no longer present, that's a neurotransmitter in case you don't know what acetylcholine is. So when that neurotransmitter is present, this channel opens, the receptor, the acetylcholine is taken away from the system, and then the channel closes. So this is going to be important in neurotransmission, but I'll leave that for neurophysiology for you. Okay, so I'll wrap it up there. I've gone on a little bit longer than I wanted to. We'll come back and we'll talk a little bit more about GPCRs and the signal transduction processes that they can trigger. I'll speak to you in a little bit. Bye for now.